Hello students, today we will study about the cell, the unit of life. It is the second chapter in your ICSE class 9 textbook. So, we will start with what is a cell. You have learnt in biology that biology is the study of all life forms. There is a huge range of living organisms from microscopic to the macroscopic, single cellular to multicellular. But all the living organisms have a basic feature in common, that is, they are made up of the building blocks called the cells. So, cells are the building blocks of the organisms. The cell is the fundamental, structural and functional unit of all living beings. It is the smallest part of the body of an organism which is capable of independent existence and of performing the essential functions of life. So, we can conclude that the cell can exist independently and also can perform the essential functions of life. Every organ in our body, whether the skin, the brain, the muscles or even our bones, are also composed of hundreds of thousands of such cells. Similarly, every part of a plant, whether the leaf, the flower, the root and even the wood, is composed of an exceedingly large number of cells. Every cell has its own life. That is, every cell has to die one day. So, every cell has to die one day. Old and weak cells in the body continuously die and are replaced by new cells. Red blood cells, for example, die after about 120 days. So, we can say that the lifespan of RBC is about 120 days. So, cells are so small, that is microscopic, that they cannot be seen with the naked eye. We cannot see these cells with our naked eye. We need an instrument for that. So, therefore, it was therefore natural that their existence could not be detected by man until he invented the magnifying aids in the form of microscopes. Most organisms, including ourselves, start life as a single cell called the zygote. That is, the zygote is also the single cell from which our life starts. The invention of the microscope and the discovery of cell. The first microscope was constructed by a Dutch scientist. His name was Antony van Leeuwenhoek. He was an ordinary public official who ground lenses and made microscopic observations as a hobby. So, his hobby was to ground the lenses and observe the things under the microscope. He is said to have constructed approximately 400 microscopes. Basically, all his microscopes were called the simple microscopes. Why they were called simple? Because they consisted of a single biconvex lens. Some of these microscopes had a considerable magnifying power up to 200 times. That is, they can magnify the image up to 200 times. Means we can see the image by increasing its magnifying power 200 times the original size. One of the Leeuwenhoek's microscope is shown in this figure 2.1. You can see in this figure, that is, a simple microscope is there and the eye is kept close to the lens on one side of the object. Right, these are the eyes and these are kept close to the lens on one side, and the object was mounted on the needle-like screw point and on the opposite side of the lens. So, Robert Hooke was another scientist. He was an English scientist and he developed a microscope by using two lenses for achieving greater magnification. Such microscopes were later known as compound microscopes. So, why they were called compound microscopes? Because the object is to be seen was placed on the stage below and light from an oil flame was thrown on it by means of a concave mirror, and he examined a thin slice of cork under his microscope. There was a box-like compartments piled up together. This reminded him of the rooms or cells of monks in a monastery. They were just like the rooms or the compartments, and so he said the cork was made up of cells. The cells which Hooke saw were all the dead cells. They were not living and they had only the empty boxes or the walls. The ordinary compound microscope, which you can see in this figure 2.3, is a greatly improved design of the original Hooke's microscope. So this microscope which you are seeing in this figure is the modern compound microscope.